Hi everyone. As you know, secretion is one of the major GI processes, and even though secretions are very important for the digestive system to function properly, you're probably starting to appreciate how secretion can also be one of the more challenging aspects of the digestive system to learn about. So in this video, we're going to take a closer look at gastric secretions. We'll talk about the specific cells inside the gastric pit, what they secrete, and ultimately how the process of hydrochloric acid formation is regulated inside the stomach. So remember that the gastric mucosa has a specialization known as the gastric pit. And this is where most of the secretory epithelial cells are located. Even though it looks like all of these cells are the same, there's actually quite a bit of diversity. So for example, we have the chief cells, which are responsible for secreting the inactive endopeptidase pepsinogen, which gets converted into its active form, pepsin, by hydrochloric acid. Hydrochloric acid is formed by parietal cells in the lumen from the secretion of hydronium ions and chloride ions. These parietal cells are regulated by histamine, which comes from enterochromaffin-like cells, by gastrin, which comes from G cells, and finally by somatostatin, which comes from D cells. Most of the other cells are mucus-secreting cells, which help to protect the stomach from hydrochloric acid. So now that we've introduced all of the major players, let's take a closer look at how acid formation is regulated. So remember just a second ago we said that it is our parietal cells which are responsible for forming hydrochloric acid inside of the stomach. This is possible because on their apical membrane, the one that opens up into the lumen, they have a chloride transporter, but more importantly, they actually have a hydrogen potassium ATPase pump, which uses active transport to pump hydrogen ions out into the lumen. This is necessary because the concentration of H+ is already very high because the stomach has a very, very low pH of about 2. So when we want to turn on this process of acid formation, we're primarily trying to regulate this proton pump because that's the rate-limiting step. And we can do that using secretions from our enterochromaffin-like cells. So remember that enterochromaffin-like cells release histamine, and histamine has a receptor on the parietal cell that's located on the basolateral membrane. So when histamine binds to its receptor, this is going to activate cyclic AMP-dependent kinases, which will phosphorylate the pump and lead to increased proton pumping. Parietal cells can also be regulated by G cells. Remember that G cells release the hormone gastrin, and like histamine, gastrin also has a receptor located on the basolateral membrane of the parietal cell. So when gastrin binds to its receptor, this will lead to the activation of calcium-dependent kinases, the phosphorylation of the proton pump, and increased hydrochloric acid formation. Now, what's really interesting is that gastrin also has receptors on the enterochromaffin-like cell. So not only does gastrin have a way to directly stimulate the parietal cell, it can also indirectly stimulate them by stimulating enterochromaffin-like cells to release histamine. So gastrin is actually a pretty potent regulator of hydrochloric acid formation. And if we think about what the ultimate effect of hydrochloric acid is to make an active endopeptidase, then it might make sense that one of the major stimuli to turn on the G cell is actually the presence of foods and amino acids inside of the stomach. Now, like most structures in the digestive system, we also have a lot of higher level nervous controls too. So, for example, our parietal cell is innervated, as is our G cell. So what this means is that just looking at food, smelling food, tasting food, or thinking about food can oftentimes lead to the release of acetylcholine onto those parietal cells and an increase in hydrochloric acid formation. We can also release gastrin, releasing peptide onto the G cells to turn on that process that way. In terms of turning things off, one thing we can always do is remove those stimuli that turn things on. So get rid of the food inside of the lumen, or remove that stimulation of higher brain centers to turn off that release of neurotransmitter. But the stomach also gives us a really good example of a negative feedback loop. So as this concentration of hydrogen ions inside of our stomach increases, that's going to lead to the activation of our final cell type, the D cell. And D cells release the inhibitory molecule somatostatin. Somatostatin has, again, receptors located on the basolateral membrane of the parietal cell. So when somatostatin binds to its receptor, that's going to decrease cyclic AMP, decrease phosphorylation of the proton pump, and turn off acid secretion formation inside the stomach.